Good afternoon, and my name is Kyota Yasuda. By the way, now is my home place, so this kind of homecoming event for me. So I'm going to talk about my research highlight. It's about the biomolecular condensation, and actually, our finding about the amino acid chirality and affect to the regulation of the condensation. So let's start from the introduction. So as we all know, we are composed of multiple types of molecules, such as like waters and peptides, polymer amino acids, and then the lipid or the sugars or nucleic acid or other things. And our specific portion of those molecules can form the cell, and then specific function group of the cells form the tissues or organs. And then a set of the tissues can form the individual live things like animals. This is an example of the zebrafish. Then, so even though we have this kind of layered structure, knowing how those molecules behave inside a cell is very important to understand how our life system is controlled. So let's looking for the cells. So this is very famous picture which is describing how the biomolecules are found inside our cells. And because cell have to be liquid to some extent, the molecules can be fluidic, and then they can move randomly basically. However, because we are very highly ordered, so we have a very highly ordered system. So there must be some system to regulate those chaotic molecules to be compartmentalized and regulated, spatio-temporally regulated. So one major system for that is using the lipid membrane and compartmentalize some specific groups of the molecules and make it very special site for some very special chemical reaction. But in the other case, several different molecules can form the condensates by their cell accumulation, which is we call the membraneless organisms because they don't use a lipid membrane, or we just say that as biomolecular condensates. So this is our target phenomenon of our and this is kind of the real image of the biomolecular condensation. They form this granule inside the cells. This is called the stress granules because they form this granule under the stress accumulation. Stress is the UV damage or the oxidative stress or the heating or something like that. And actually, this formation do not need cellular circumstances. The purified protein of the component of that granule can form this like a liquid droplet by themselves. In this case, they can form this droplet by increasing the concentration of the protein. And so what they are doing is, many reports are saying different functions of those, like they can do. They can be a reactive vessel, so they can accumulate something and then export something, or enrich or localize something, and then sometimes buffering something or inactivate something by sequestering some specific molecules, or can be part of the sensing system. So many studies has reported that main driving force of this droplet formation is the liquid space separation of the biomolecules such as a protein and a nucleic acid. And these are kind of representative interaction systems that are used in this kind of condensation, such as biomolecular recognition. That is kind of structure-based recognition, or ligand binding is also basically the structure based on interaction. And the others are like charge-based interaction, which is usually brought about by the feature of the amino side chains, like if tyrosine or tyrosine origin or asparagine or something like that. So our group is kind of trying to figure out the underlying mechanism which control the condensation of biomolecules. And then I will show the three different targets which is kind of relating to the disease actually, so that you can imagine why that's important for us to study on it. So one is the virus replication. Our in a collaboration with Dr. Chayama's group in the Kyota University found the protein which is binding to the human hepatitis B virus capsid protein. And we found that this found protein can form the condensate like this, and they incorporate the capsid protein inside a droplet. And then when we see these samples with the electron microscopy, we can see the increased amount of the virus capsid structures. So this ring-like structure is a virus capsid. So that means, I mean, depending on the condensation, they increase the number of capsids. So that means, virus may be using the host protein and the human protein as a factory to form the capsid efficiently. And in other words, if we could manipulate the condensation of those proteins, that might be a new novel therapeutic way for the hepatitis B virus infection. And the other case is about the neurodegenerative diseases. This is kind of example of ALS, amyotrophic lateral sclerosis. Actually, not only the ALS, but many other neurodegenerative diseases have a hallmark of this kind of protein aggregation. And then this kind of protein aggregation is now known to be, caused by the phase separation of the subproteins, and then, aggregation of those condensates are probably the basic cause of this kind of neurodegenerative diseases. And then the other case is about the cancer cell. So as I shown in the movie, the stress gradient is found under stress for the cells. And then the anti-cancer drug is also stress for cells. And then the cancer cells are known to form stress gradient more than the normal cells. And then they use the stress gradient as kind of, the way to reduce the anti-cancer drug. And we found that one of the component protein of the stress gradient knockdown of those protein reduced the stress gradient number. And actually, in those kind of cells, when we treat those cells with the anti-cancer drug, they show more of the off cells. That means the stress gradient number is really important to be resistant to the anti-cancer cells. So I hope you imagine why those biomolecular condensation is important for our life. But today in this talk, I'd like to talk about the enantiomeric effect of the amino acid about the biomolecular condensation because of the SKCM² issue. So as people in the forward talk about the amino acid has chirality. This is just an example of how. And because the protein is composed of L types, are usually mainly composed of the L types of the amino acid, and so that the different chirality may affect to that the protein polymerization as like a major effect. So that's idea. Different chirality may perturb the condensation differentially. So first case of the data of us is about the FAST protein, which is one of the genetic cause of the ALS. So as I showed, that FAST protein can form the aggregate inside of the patient's tissue, and then they can form the desired droplets. So they have this kind of structures, and they're rich in the tyrosine and arginine, and this tyrosine arginine function is known to be the main driver of the condensation of this protein. And actually, excess amount of tyrosine arginine can perturb the formation of the droplet. And what we found is that the difference in chirality can affect the different level of the perturbation of the phosphoprotein condensation. So this is a cell expressing the phosphoprotein and forming things like granules inside the cells by phase separation and condensation. And when we inject the tyrosine, an L-type or G-type of tyrosine, they do actually reduce the size of the granules. And this is kind of a tracing of the relative size differences. And we found that D-tyrosine is more effective to, reduce or to reduce the size of the granules. So that means there's some differential effect of the amino acids to the condensation depending on the chirality. And this is another case. This is a protein which is component of the stress granules. Then, the condensation show that it's like tolerability. I hope you can see the differences. If there is no condensate, it's very clear. However, if it's once it's condensed, that show the tolerance problem. So by using this feature, we can measure the turbidity to other indicator of the condensation. And then we found that increasing number of the G-type of tyrosine can reduce the turbidity. That means that can prohibit the condensation of the stress granule component protein. So, and then I just show the example of tyrosine, but many other types of amino acids show the similar effect. That means this effect is not depending on the feature of side chain, but the chirality itself is important to perturb the condensation. So we check how such phenomenological data. So I don't know what is actually happening in molecular levels. However, this is kind of a nice starting point, I think. So in summary, we can say that chirality dependent effect exists in the perturbation of the biomolecular condensation. And however, we don't know what is happening actually. And the other thing, this is kind of very much of my personal interest, but manipulation of condensates by chiral dependent effect has a very important implication for the novel therapy, right? For such a very severe disease for human. So I have to thank to our group members and collaborators and our clients. Thank you for listening.